All right. So conjugation is something that's going to primarily occur with gram-negative bacteria. And it can account for the transfer of a large quantity of DNA. But in this case, there must be contact between the donor of the DNA and the recipient in order for that transfer to occur. So right here, this bacteria, we can't tell from this which one is called the F plus cell. The F plus means it is positive for that F plasmid. And F actually stands for fertility. So the F plus is going to be the plasmid containing cell, and the F is the fertility plasmid that's going to be responsible for making the pilus. So the recipient cell will be an F minus cell, does not have the plasmid to make the pilus, and the donor cell will be an F plus cell. All right, and when donor cells and recipient cells are mixed together, those pilus bridges form between the two, and the F is actually transferred. Okay, so the F plasmid is transferred through that conjugation pilus. Once that is actually transferred, then the second cell will become an F plus cell as well. So let's look at what's happening here. First of all, this has an F plasmid. This can form the conjugation pilus, and it'll form the pilus and it'll pull the cell closer to it, and then our F plus cell is here. Notice this doesn't have a conjugation plasmid, an F plasmid, so it's an F minus cell. And as this pulls it closer, remember this is double-stranded, right? Double-stranded DNA. This one strand of that will go through the conjugation pilus into the F minus cell. And as that's happening, this will be made whole again, right? We will make it double-stranded again. And as this one strand's coming over here, it will be made double-stranded as well. So now we end up with that pilus in both of these. Now this pilus, or this cell, is an F plus cell. It means it can pass that plasmid to other F minus cells in this same process. I think I'm going to end here, so you don't need to, no, I guess we'll do this, sorry. This is called high frequency recombinations, or HFRs. And this term is used to recognize cells that instead of having their F factor as a plasmid, they're still an F positive cell. They have the fertility factor, they have that, but the plasmid has been incorporated into the bacterial chromosome. It can go in and out of the bacterial chromosome, but at portions of its life, it is within the bacterial chromosome. So when the bacteria itself undergoes binary fission, it is replicated with that cell and goes to both daughter cells. When an HFR cell is the donor in conjugation, sometimes genes next to the plasmid can be transferred. And I do have a picture to show this, but genes are transferred in a linear sequence starting at the origin, and the DNA replication occurs by rolling circles. So one strand is retained and one is sent to the recipient. But remember now, we're talking about not just the fertility plasmid, we're talking about the bacterial DNA, because the HFR cell has that plasmid incorporated in the DNA. So the bacteria now has the ability to make that pilus, but instead of just sending over that one plasmid, it has the potential to send over on either sides of that plasmid bacterial DNA. Now, the plasmid does not occur, the plasmid does not last forever. The bridge does not last forever. So when this happens, whenever that breakage occurs, will determine how much of that DNA is actually transferred. So interestingly enough, with an HFR cell, usually the F negative cell to which that plasmid was being transferred usually remains negative, because the F factor frequently does not get all the way over there. And that would mean that it would remain an F negative. That separation of that particular plasmid will occur because the bridge gets broken. It would take about 100 minutes for the entire genome to transfer, and frequently the bridge is broken long before that time. So let's look at how this works. This is our donor chromosome, okay, we can see here. And our this is our F plus cell. And in this case, now maybe, and it looks like we just got the F plasmid in this cell, right? Because here's a pilus. So we got the F plasmid from another cell, and it's incorporating into the bacterial genome. So now it's in the bacterial chromosome, and now it's an HFR cell because it's incorporated into the bacterial chromosome. It can still make a pilus with an F negative cell. So it's made this conjugation bridge, or pilus, or sex pilus. There's a number of names. But now it needs to transfer, because it's made this pilus, it wants to transfer this F. But it's going to start at an origin. It does not have to start right where the plasmid is. There are certain origins of transfer. It's going to start at one origin of transfer, and look at it, it's going to start bringing this over. In this case, the origin of transfer, it looks like, took a little bit of the F plasmid, and then it's going this direction. And as it's going this direction, it's being made double-stranded. This is being remade. But as it goes across here, once this bridge is broken, whatever has gotten across of that F plasmid is what's going to be there. So now we have mostly bacterial genome and only part of the F plasmid. The rest of the F plasmid remains in the bacterial genome of the original cell, the HFR cell. So we get done here, and we can incorporate these new pieces of bacterial genome into the recombinant cell, or the new cell that we've made. But notice, even though we've got some of that F plasmid, we never got the entire F plasmid. So this cell is still an F minus plasmid, or an F minus cell.